This method can help answer key questions in the cardiac electrophysiology field about the structure of the extracellular space and cellular communication mechanisms. This technique's main advantages are that it has high throughput capabilities and an improved spatial sampling frequency. Essentially, we can obtain measurements more quickly and with higher confidence than before. Paranexus identification and program troubleshooting are difficult to learn without visual demonstration because the paranexus is a relatively newly defined structure, and troubleshooting may not be intuitive for investigators who are unfamiliar with MATLAB.
For grayscale images, use numerical computing software to ensure that no pixel has an intensity greater than 255. Then, open a prepared image in image processing software and zoom in on the paranexus.
It is critical to properly identify the paranexus. The first step is to identify a gap junction plaque which has a striped appearance. Then, we look for two opposing membranes in plane out to about 200 nanometers.
Identify the gap junction plaque from its pentalaminar structure. The beginning of the paranexus is the point at which the two opposing cell membrane bilayers diverge. Display a scale bar in nanometers. The starting and ending points of the paranexus outline will be 200 nanometers from the beginning of the paranexus. Next, select the freehand selection tool. Click and drag or use a stylus to carefully trace up along the inner membrane of one cell to the beginning of the paranexus, and back along the inner membrane of the second cell. Close the selected area by releasing the mouse button or lifting the stylus. Then, set the line width to 1 pixel and the foreground color.